Previously we have used the term ganglion. So let's describe what ganglions are. Dorsal root ganglia, which we have termed before, is a collection of cell bodies of pseudo-unipolar neurons. In this area, which is running next to the spine, you do not have synapses. One way to identify this particular ganglion is to look at the cell body and see the placement of the nucleus, which is centrally located. Around the cell body, you also notice there are cells which are referred to as the satellite cells, which are a type of glial cells. In this picture, it becomes a little clearer. So here you are seeing the cell body of a pseudo-unipolar neuron with these satellite cells, which are clearly depicted surrounding the cell bodies. The function of these satellite cells is to control the local microenvironment, is to supply the nutrient to the neural cell body as well as structural support. In addition, it also provides protection to the neural cell body by providing cushioning. So on the other side of the dorsal root ganglia, we have these autonomic ganglions, sympathetic and parasympathetic. In this case, the cells present here are multipolar neurons. Easy way to identify them is to look at the location of the nuclei, and you will notice that they are eccentric. Here the synapses do occur, and there are satellite cells surrounding these cell bodies, but they are fewer. They also contain these lipofuscin granules, which are nothing more than sort of like a lysosomal granules or vesicles within the cell body. And in here, the nerve fibers tend to be unmyelinated. So in the parasympathetic ganglion, and you have seen this in the GI tract, for example, you have seen the outer and the inner muscularis layer. There was aggregation or collection of these neurons, which are referred to as the parasympathetic ganglia. So moving a little bit more in detail about the peripheral nerve itself, one thing to remember is that peripheral nerve is an organ by itself. So it has cells of the neuron component as well as connective tissue blood vessels as well. So this diagram depicts that in an overview. So if you were to look at that in more detail, what we will be looking at in the lab, what you notice is peripheral nerve contains a hierarchical structure where you have the nerve axons themselves, which are surrounded by what is referred to as the endoneurium. Further up, the collection of these axons is referred to as a fascicle, and that is surrounded by connective tissue referred to as the perineurium. Collection of these fascicle, which forms the actual nerve, is surrounded by another set of connective tissue is referred to as the epineurium. And here you can see the sensory neurons as well as the motor neurons are running within that whole big nerve, as well as autonomic unmyelinated neurons. This is a histological view, and you can see the bundle here of the nerve fibers within which you have individual axons. So if you look at the whole collection here, so again going from hierarchical way, you have the endoneurium, which isn't labeled here, surrounding individual axons, followed by a perineurium. In a higher view, you can see that is surrounding each one of these fascicles, followed by a hierarchical structure of epineurium, which is surrounding multiple fascicles, which are listed here, BNF here, here, and here. In a longitudinal section, you see a slightly different view and maybe more informative view. First thing that you will notice is that nerves tend to have more of a wavy pattern, which is more clearly visible in this higher power view. Myelin is more prominent, and for that reason most of the structure is paler in color. So M is the myelin, and this is the axon running within it. And you also see here node of Ranvier. Also visible are the nuclei of the Schwann cells. So all in all, this is a nice depiction of what a peripheral nerve looks like in a longitudinal section. In the nervous system, there are lots of sensory elements involved. There are receptors all over the body which are sending signal back to the CNS. So they are physical stimuli which are being passed on to the central nervous system. They encode a number of information such as what's the nature of that stimulus, the location, the intensity, as well as the duration. Cell bodies for sensory receptors reside in the DRG, and the information enters there through the dorsal horn. Axons transmitting these very fine discriminatory touch as well as proprioceptions are highly myelinated. They need to be allowed to conduct the impulses at a fast rate. On the other hand, those impulses coming, sensory impulses coming for pain and temperature are unmyelinated. For that reason, they have a slower conduction velocity. So let's spend some time looking at peripheral nerve injury. This is an important part of tissue regeneration as well as clinical correlations. You can classify peripheral nerve injury into either a mild compression, where there is no real demyelination of the axons and the recovery is pretty normal, followed by crush injury, where the axons within a nerve fascicles can become disconnected, followed by a more severe injury, which is referred to as complete transection of the entire nerve, and injuries such as, for example, trauma from blade entry or a ballistic entry. So what does happen when a nerve gets injured? In this diagram here is being depicted as in a relatively normal neuron innervating a muscle here. When the injury happens, there are two things that happens. Events take place both anterograde and retrograde. The anterograde events are the degeneration of the axon from the site of injury to all the way to where the nerve is innervating the tissue. In this case, there is a muscle innervation, and what's happening, if you look at this picture here, the muscle is atrophy. It's becoming weaker and smaller in size. On the other hand, on the retrograde events, the cell starts to swell up. There is a chromatolysis that's happening in the proximal part of the cell body. You notice here, for example, as well as there's a shift of the nucleus from the central to the peripheral site. There's also a breakdown in the RER or Nissl bodies. So there's a lot of things that are happening inside the cell body as well as the axon. In addition, if there's a lot of damage here and the exoplasm, which is the axon cytoplasm, is severely compromised, then the chances are this neuron will die. During regeneration, the whole event has to be reversed. here, if the regeneration is to happen, the injury has to be limited. One thing to note is that regeneration is possible in the peripheral nervous system, but not in the central nervous system. So when it does in the peripheral nervous system, when the injury is regenerated, the Schwann cells, which from the site of the injury, starts to proliferate, and they line up to form these guidance tubes towards the target organ. And this is where the cells start to produce these neurotropic substances which are guiding the regenerating axon. Overall, the regeneration rate is very, very slow. The regeneration, when it does happen, it can be seen here, for example. It is depicted as the axons then re-innervating the muscles or target organs, and is seen in this picture here. So as said before, that during the nerve regeneration, the neurites are sprouting, which contain that growth cone towards the target organ. The rate is slow, considered to be between 1 to 4 millimeters per day, which is very similar to the axon transport that we saw before, which was around 1 to 4 millimeters per day. So you can imagine that the regeneration itself is dependent on how well the material or the transport that can happen to the end of that regenerating axon. To help that repair, sometimes you have to bring the ends of the nerves together, or superimpose some kind of a graft on top of the nerve injury, and in doing so you're providing that matrix for continuity, and that encourages the regrowth of nerve. And that's a common, nerve grafting is a common procedure for large injuries. However, you are relying on individual axons making perfect connections from the cell bodies to the target organs. If there is a mismatch between the ends, then you may lose specificity and or full recovery of that target organ.